Hello, my name is Amber Notah. I am a senior here at the University of Arizona. I am majoring in biology and I'm going to get an emphasis in biomedical sciences, so I will be continuing on another year. I currently work in Dr. Gerardo Lopez's lab and we are researching the parasite Cyclospora cayetanensis. And just give me a second while I pull up my poster. Okay, here we are. And I'll first start off by saying that this research is to understand the prevalence and transmission of Cyclospora cayetanensis in the West Coast. And I'll give you a little background on our research as well as the parasite Cyclospora itself. So Cyclospora cayetanensis is a coccidium protozoan parasite that causes cyclosporiasis, which is a worldwide diarrheal disease. The illness cyclosporiasis is normally associated with consumption of fresh produce or water that is contaminated with the oocyst from the coccidium protozoan parasite. The symptoms of this illness include diarrhea, cramping of the muscles, weight loss, bloating, influx of gas, loss of appetite, fatigue, and nausea. Now, it's not definitively known how fresh produce or water gets contaminated with the parasite Cyclospora cayetanensis. We know that this parasite can only infect other people when its oocysts have been sporulated in the environment, then ingested in order to complete its life cycle in the human host. This means the transmission route of the parasite is through fresh produce or water contaminated with oocysts that have become sporulated between 1 to 14 days. C. cayetanensis in fresh produce has become a major issue in endemic areas of a plethora of countries throughout the world. And as of recently, it has become an issue with produce grown in the USA. In this study, we want to gain a better understanding of the prevalence and transmission routes of C. cayetanensis in environments where fresh produce is grown in the West Coast USA. To expand our knowledge, this study consists of collecting samples from produce growing regions of the West Coast. The samples currently collected include irrigation and surface water, swabs of portable toilet surfaces, and rinse water of the portable toilet stations. These environmental samples are then shipped to the University of Arizona, where they are processed and analyzed using the FDA BAM Chapter 19C methods. Briefly, the recovery of oocysts from filters through a backflushing process, DNA extraction and purification, and molecular detection by real-time PCR that targets the C. cayetanensis 18S rRNA gene. So that was just a little bit of background on the parasite as well as our research that we're doing at the U of A. And now I'll move into the outbreak. So according to the CDC, as of September 28, 2021, 1,020 laboratory confirmed cases of cyclosporiasis have been identified. At least 70 people have been hospitalized, but 0 deaths have been reported. And in this little map of the United States, all the green states that are shaded are states that have cases associated with them with cyclosporiasis. And for our objective for our research, it is to detect and determine the prevalence and transmission routes of Cyclospora cayetanensis in fresh produce environments from the West Coast of the United States. And now I'll move into our methods. So like I said, some of our samples will be shipped to us to the University of Arizona. And for the West Coast region, these get shipped to us directly. We don't go to the West Coast and collect them ourselves. We'll just do all the lab work. And so first we'll have our sample collection, which includes the portable toilets, the swabbing of them, as well as the rinse water that they collect for us in a little jar that we call a sludge. And then they filter the water through our Rexy 25S filters. And then after that, they get shipped to our lab and we'll start a backflush process on them. So our second picture will be our backflushing. And then our third, once we get the extracted water from the filter through our backflush, we'll put that into our centrifuge in order to get a pellet. And then from there, once we get our pellet, we'll put it through our flat sieves, and this will just help us with the flow cytometry. That's the next step. And then after the flow cytometry, we'll go into microscopy because we want to see if we can actually see the Cyclospora cayetanensis, because there's a little fluorescence with it, and that's what we want to identify. Then once we finish with the microscopy, we'll move into the DNA extraction and purification. Then we'll go into the qPCR test. And from there, we can do genome sequencing on our sample. And so for our results, I put together a little table. For the first column, we have the type and amount. And for the different types that we have from the West Coast that we receive are 3 types. And this is the filter itself from the water that they filter through. And then we have the porta potty samples. And then we have the sludge. So we have right now 27 filters, 24 porta potty samples, and 5 sludge samples. This brings it to a total of 56 samples here in our lab. And a total of 56 samples have been received from the West Coast growing regions of the United States. And these samples are still currently being processed through our methods. Now, since this is just a small part of the bigger research project that we're doing, we have so much samples coming in from different areas of the United States. So all of our samples are still being processed. And this research project will just help us understand the prevalence and transmission of cyclosporic cayetanensis in produce growing regions of the West Coast of the United States. With the variety of sample types that we have collected/received, we can identify potential routes of C. cayetanensis to fresh produce. Using flow cytometry will help identify serotypes and good sample quality for genome sequencing. And I just want to give a couple of acknowledgements to Dr. Gerardo Lopez, who allows me to work in his lab, and Cecily Citadel, Dominic Rodriguez, Chi Nugent, Bronson Gould, Tommy Jody, and Janelle Cartez. I'd also like to thank WASO for giving us these opportunities for underrepresented students, as well as the School of Animal and Comparative Biomedical Sciences and the University of Arizona. And so lastly, here are all of my references. We mostly use the Almeria paper, Sonar, and Kivarstrom, and I also have the CDC reference right here down at the bottom. If you have any questions or anything, please let me know, and I will try my very best to help you understand them. Thank you. Bye.